There's a tube inside the human body that, if fully extended, would measure between 6 and 7 meters long. It fits neatly folded within an abdominal cavity that's barely 30 centimeters deep. But that's not the most surprising thing. What's truly extraordinary is that the internal surface of that tube, if fully unfolded, would cover an area of approximately 250 to 300 square meters, a surface the size of a tennis court contained within a person's body. That's the anatomy of the small intestine, a feat of biological engineering that has been fueling every thought, every movement, every heartbeat of humankind for millennia. For centuries, anatomists viewed the small intestine as a simple conducting tube. Galen of Pergamon, in the 2nd century, described it as a passive channel through which food passed before reaching organs he considered more important. For over 1,000 years, this view prevailed. It wasn't until the Renaissance, with the work of Andreas Vesalius, and later with the microscopes of the 17th and 18th centuries, that scientists began to suspect something different. What appeared to be a smooth, uniform membrane turned out, upon closer inspection, to be an architecture of almost unimaginable complexity. Every square centimeter of that inner tube concealed structures that defied any analogy with human engineering. Today we know that the small intestine is not simply where nutrients pass through. It is where nutrients are built, recognized, selected, transported, and converted into fuel for every cell in the body. In functional terms, it is the organ that determines whether the body thrives or not. To understand why, we must begin with its most basic structure, its internal topography. The small intestine is divided into 3 segments with distinct yet continuous functions. The first is the duodenum, which is only 25 centimeters long, but represents one of the most active biochemical crossroads in the body. Here, pancreatic juices, rich in digestive enzymes, and bile, produced by the liver and stored in the gallbladder, converge. This combination transforms the partially digested contents arriving from the stomach, known as chyme, into a chemical mixture that can be absorbed. The duodenum doesn't absorb much on its own, but it prepares the way for the 2 segments that follow with a biochemical precision that no external laboratory has been able to fully replicate. The second segment is the jejunum, which occupies approximately 2/5 of the total length of the small intestine. If there is one place in the body where nutrient absorption occurs most intensely, it is here. The jejunum is densely equipped with structures that multiply its functional surface area, and its cells work at a speed few other parts of the body can match. The third segment is the ileum, which comprises the last 2.5 or so, and specializes in absorbing components not fully processed by the jejunum, including vitamin B12 and bile salts that will be recycled and returned to the liver. But the division into 3 segments, while useful as a map, still doesn't reveal the secret of the small intestine. That secret lies in its microscopic architecture. When the inner wall of the small intestine is examined under a microscope, a structure appears that anatomists called circular folds, or plicae circulares. These are permanent folds of the mucosa, the innermost layer of the intestinal wall, which do not disappear even when the intestine is distended. These folds already triple the available surface area, but on top of these folds is another structure. Millions of tiny projections called intestinal villi, or simply villi. Each villus measures between 0.5 millimeter and 1.5 millimeters in height, and is shaped like a finger or a leaf. They are vascularized structures. Each one contains blood capillaries and a central lymphatic vessel called a lacteal, which plays a crucial role in fat absorption. Villi increase the functional surface area 10-fold compared to folds, and we haven't even reached the smallest level yet. On the surface of each cell lining the villi are even smaller structures called microvilli. These are cylindrical projections, barely a micrometer high, so tightly packed together that they form what cytologists call the brush border. Under an electron microscope, this border looks like a dense, even carpet. Each intestinal cell has approximately 1,000 microvilli on its apical surface. This level of magnification adds another 20 to the total surface area. The cumulative result of all these folds, villi, and microvilli is that seemingly impossible figure: 250 or 300 square meters in a 7-meter-long tube. The evolutionary reason for this architecture is not difficult to deduce. Organisms that absorb nutrients more efficiently survived better in environments where food was scarce or unpredictable. The multiplication of surface area is not a whim of biology, but a direct response to the selective pressure of millions of years. A small intestine with a larger surface area absorbs more nutrients per unit of time. In times of scarcity, that difference could be the difference between survival and death. However, a large surface area is worthless if the cells lining it aren't equipped to do their job. The cells of the brush border, called enterocytes, are some of the most active in the body. They are completely renewed every 3 to 5 days, meaning the small intestine rebuilds itself several times a month. This rate of renewal makes it one of the tissues with the highest rate of cell proliferation in the adult body. The energy this process requires is considerable and reflects the organ's vital function. But the enterocytes don't work alone. The brush border is covered with digestive enzymes that complete the hydrolysis of nutrients: sucrase, maltase, lactase, and peptidases. These enzymes break down disaccharides into simple monosaccharides or short peptides into individual amino acids just before they are absorbed. Absorption is not a passive process. Monosaccharides like glucose and galactose are actively transported into the cell using sodium-coupled cotransporters, specialized membrane proteins that consume energy in the form of an electrochemical gradient. Fructose, on the other hand, enters by facilitated diffusion through a different transporter. Amino acids have their own transport systems, several of which are also sodium-coupled. Fats follow a different pathway after being hydrolyzed into free fatty acids and monoglycerides by pancreatic lipase. They associate with bile salts to form structures called micelles, which allow them to reach the enterocyte membrane. Once inside the cell, the lipids are reassembled into triglycerides and packaged into structures called chylomicrons, which are then released into the lymphatic lacteal of each villus, thus bypassing the hepatic portal system, at least on their initial transit. The fat-soluble vitamins A, D, E, and K follow this same lipid pathway. This distinction is not trivial. It means that dietary fats do not follow the usual nutrient route, but instead enter the bloodstream via the thoracic duct and eventually reach the liver, rather than passing directly through it. This detour has important metabolic consequences and is part of why the type of fat in the diet matters as much as the amount. The small intestine is also where the body does something few people associate with digestion. It monitors what goes in. The gut immune system accounts for an estimated 70% of all immune tissue in the human body. Peyer's patches, which are accumulations of lymphoid tissue distributed mainly along the length of the intestine, constantly monitor the intestinal contents for pathogens. Dendritic cells can extend projections through the intestinal wall to directly sample the luminal contents. Secretory immunoglobulin A antibodies are released into the intestinal lumen to neutralize bacteria and viruses without triggering a full-blown inflammatory response. This surveillance is continuous, silent, and absolutely essential. The gut is in constant contact with the outside world through food and must constantly distinguish between what nourishes and what threatens. Maintaining this balance between nutrition and immunity is not always easy. In conditions of chronic inflammation, alterations in the gut microbiota, or direct damage to the mucosa, the permeability of the intestinal epithelium can increase. This phenomenon has been described as intestinal hyperpermeability, or, more colloquially, leaky gut. Research into its causes and consequences remains an active area. There is evidence suggesting that increased intestinal permeability can facilitate the passage of bacterial antigens into the bloodstream, triggering systemic immune responses. However, there is still no full consensus on which diseases this permeability causes, or simply correlates with. Researchers continue to explore the relationship between the health of the intestinal mucosa and diseases that seem far removed from the digestive system, from autoimmune disorders to metabolic conditions. The microbiota inhabiting the small intestine, although less dense than that of the large intestine, also exerts an influence that scientists are only just beginning to map. The bacteria in the small intestine compete with potential pathogens for space and resources. They produce certain vitamins, such as vitamin K and some B-complex vitamins, and modulate the local immune response. Studies in animal models and increasingly in humans indicate that the composition of this microbiota influences the efficiency of nutrient absorption, the regulation of appetite through intestinal hormonal signals, and possibly the communication between the gut and the brain via the gut-brain axis. This axis involves the vagus nerve, hormonal signals, and neurotransmitters produced in the gut, including serotonin. It is estimated that between 80 and 90% of the body's serotonin is produced in the enterochromaffin cells of the intestine. Intestinal serotonin does not cross the blood-brain barrier, but it exerts local effects on motility and signaling of the enteric nervous system. Its dysfunction has been associated with disorders such as irritable bowel syndrome. The enteric nervous system deserves special mention in any discussion of the anatomy of the small intestine. It contains between 100 and 500 million neurons distributed in two ganglion networks: the myenteric plexus of Auerbach, located between the muscular layers of the intestinal wall, and the submucosal plexus of Meissner, located beneath the mucosa. This nervous system can operate autonomously, coordinating peristalsis, mixing reflexes, and secretion, without needing constant instructions from the brain. Because of this functional autonomy, some researchers call it the second brain, although the metaphor has its limitations. What is certain is that the small intestine is not a passive tube controlled from the outside. It is an organ with its own neuronal integration capacity that perceives, evaluates, and responds to its environment in real time. The motility of the small intestine also deserves attention. Peristalsis, the wave-like movement that propels the contents along the tube, is the best known. But there is another movement, less well known and more important for absorption: segmentation. Segmentation consists of rhythmic contractions that repeatedly divide and churn the chyme without propelling it forward. This movement maximizes the chyme's contact with the absorptive mucosa and mixes the contents with enzymes and bile salts. Without segmentation, simple peristalsis would transport nutrients too quickly, reducing contact time and, therefore, the efficiency of absorption. Between meals, when the small intestine is relatively empty, a different motor pattern called the migrating motor complex operates. This complex travels through the intestine in regular waves every 90 to 120 minutes, clearing away waste, dead bacteria, and mucus, and preventing the buildup of material that could promote infection. This cleansing movement is also partly responsible for the sounds the abdomen makes when the stomach is empty. Some diseases, by contrast, highlight just how central the small intestine is to life. Celiac disease is perhaps the most instructive example. In people with this condition, the ingestion of gluten, a protein found in wheat, barley, and rye, triggers an immune response in the lining of the small intestine that progressively destroys the villi. The absorptive surface area decreases, and nutrients are not absorbed efficiently. The result is a cascade of deficiencies affecting virtually every system in the body. Anemia due to lack of iron and folate, osteoporosis due to calcium and vitamin D deficiency, peripheral neuropathies, and infertility. Celiac disease is not, in essence, a digestive disorder. It is a disorder of the small intestine's architecture, and its consequences are distributed throughout the body. Because nutrient absorption is a function that supports all other organs. Short bowel syndrome is another case that underscores the organ's indispensability. It occurs when a significant portion of the small intestine is surgically removed, whether due to necrosis, tumors, or Crohn's disease. The consequence is insufficient absorption that may require indefinite parenteral nutrition. Today, small bowel transplants represent one of the most complex surgeries in modern medicine, with significant rejection rates and a mortality rate that remains high compared to other solid organ transplants. The difficulty of replacing the small intestine, whether by surgery or transplantation, is itself a measure of its functional complexity. Current research is exploring avenues that a generation ago would have seemed like science fiction. Intestinal organoids, three-dimensional structures grown in the lab from intestinal stem cells, replicate the architecture of villi and crypts with remarkable fidelity. They are used to study diseases, test drugs, and, in a future that researchers describe as promising but still distant, potentially repair or replace damaged intestinal tissue. Studies of the gut microbiome are revealing connections between the bacterial composition of the gut and diseases ranging from obesity to depression and the effectiveness of chemotherapy treatments. Understanding the gut-brain axis is transforming how neuroscientists and gastroenterologists think about mental health. There is ongoing research into how enterocytes regulate systemic inflammatory responses and the role of Paneth cells, a specialized cell type in the intestinal crypts, in defense against pathogenic bacteria. Questions remain unanswered. There is no complete consensus on how the architecture of the small intestine varies among individuals and what effect this variation has on metabolic efficiency. Research is underway to determine whether chronic subclinical damage to the intestinal mucosa caused by inflammatory diets, alcohol, non-steroidal anti-inflammatory drugs, or chronic stress has measurable systemic consequences. Research continues into how the circadian rhythm regulates the expression of transporters and enzymes in the gut, which could partly explain why the time of day a meal is consumed influences its metabolism. What is certain, however, is this. The small intestine is not the modest tube that ancient anatomists described. It is the most sophisticated interface between the external world and the internal biology of the human body. Every meal a person eats is processed by an organ that has taken millions of years to evolve into its current form. An organ that folds 300 square meters into the space of an open hand, that communicates with the immune, nervous, and endocrine systems simultaneously, that renews itself every few days, that decides what happens and what doesn't, what nourishes and what threatens. There is something profound in the idea that a human being's life, all its neurological, emotional, and muscular complexity, ultimately depends on what happens inside a silent, folded tube in the abdomen. Every thought, every act of will, every heartbeat requires nutrients that the small intestine decided to absorb. The brain that reads and understands these ideas is partly built from molecules that cross the brush border of the enterocytes. There is no hierarchy among the organs of the human body, but if there were, the small intestine would have a strong case for claiming its place at the top. Not because it is the best known or most studied organ, but because without it, all the others would cease to function. And that invisible dependency, that silent architecture the body carries within it, without anyone paying much attention, is perhaps the most honest way biology reminds us that everything we are must first and foremost pass through our inner workings. For millions of years, the human body silently perfected one of its most extraordinary creations, and today, it knows its secret. If this journey through the small intestine has taught you something new, something that changes the way you think about your own body, let us know with a like. And if you want to continue discovering the hidden architecture that keeps you alive, subscribe to Anatomy Revealed. Every week, a new structure.